My lab studies ALS, which is also known as amyotrophic lateral sclerosis, or Lou Gehrig's disease. Patients lose their motor neurons, which are the nerve cells that are responsible for actuating movement. And so they eventually become completely paralyzed and they lose the ability to even breathe. Many laboratories have been working on ALS over the past several decades. More recently, people have been using animal-based models, rodent models of the disease to try to understand what happens. There are a few limitations with it. One is that the only available animal model corresponds to about 1% of all patients. The other thing is that researchers have had to exaggerate the genetic mutation by over-expressing it in order to make the animal have ALS-like symptoms. So our approach has been different. We use stem cells that we generate directly from the patients. What we can do is then convert these cells into the motor neurons, which are the cells that are affected in ALS patients, and they retain all of the genetic information of that ALS patient. So they recapitulate the disease in a petri dish, so it's easy for us to study, and we can perform many experiments and hopefully figure out how the disease works in a more accurate way than we ever have before.